Sounds
Hello,
good.
everyone. Welcome to the Thomas Expert Series. Thank you for joining us today. We're going to get started in just a minute. In the meantime, I want to double check, make sure everyone has their microphones muted. This expert series will be recorded and placed on demand within 24 hours, so please let your colleagues know how much you enjoyed the presentation today and direct them to the on-demand tab or have them sign up for future talks. To get everyone a little bit more familiar with the chat, where is everyone tuning in from? I'm in New Jersey. Michael, where are you at?
I am in New Jersey.
Awesome. So we've got a couple New Jerseys in here in the chat too. We've got Woodbury, New Jersey, Ocean City, New Jersey. We've got Florida, more New Jersey, Texas, Colorado, Ohio, more New Jersey, Seattle, San Diego. So all right, so we will be holding a Q&A after the talk, so please hold your questions off until the end. And at this time, I'll go ahead and turn it over to Michael here to talk about supporting full workflows for cell and gene therapies.
Thanks, Heather. Thank you, everyone, for joining me today. I'm Mike Moeller, uh, and I am the Director of Business Development for Cell and Gene Therapy here at Thomas Scientific. Just to introduce myself a little bit and tell you a little bit about my background, I've been operating in and around the fields of cell and gene therapy for a little over 25 years now. I've actually been a scientist for 37 years, but I really got into cell and gene therapy about 2.5 decades ago. I've worked in a variety of capacities. I've worked on two different cell therapies, one of which is in clinical trials right now. So we're keeping our fingers crossed with that becomes a therapeutic. Also was part of a team that worked on one of the very early gene therapies. So I've been on both sides of the fence, cell and gene therapies. I have been a university professor. I have also been the chief scientific officer of a biotech. I've been the scientific director of a CRO. So I've had a lot of experience in this field. And the biggest reason I came to Thomas was really because I'm sort of at a stage in my life when I want to support this field as I see it grow. I was kind of doing cell therapy before cell therapy was cool, as they say. And I've watched it grow up around me, and I've been delighted. One of my hobbies is gardening, and I feel like I've watched a lot of flowers bloom, and I take a lot of pleasure in helping Thomas Scientific to kind of get where they're going. Um, I am helping them to basically build out their portfolio to also home in a little more precisely on cell and gene therapies and bring products and services to the community that are useful, things that will basically help the field improve and move ahead even faster. Seems like it's already moving ahead so fast, but um, and lastly, the thing that I like to tell people is I am here to support you, the customers. I am here to make sure that you succeed. Like I said, this is a labor of love for me. This is something where my heart and my soul are in this stuff. So I'd like to talk to you a little bit today about what I call the new Thomas Scientific. And I say the new Thomas Scientific simply because there are a lot of new and exciting directions that we're moving into. We've traditionally been lab consumables, and we still do that very, very well. But we're moving into some exciting areas. And I can tell you we're really, really, really excited as a team about our efforts in cell and gene therapy. We all kind of recognize, especially, I'm sort of the hype man for cell and gene therapy here at Thomas. And we realize that these are the medicines of the future. These are things that are going to change people's lives. These are things that are going to save people's lives. They give quality of life to people. They're going to give hope. These things have meaning. These things have value. And so we want to do our part to make those miracles happen. And we want to bring value to you. Now, before I get going, I'm going to turn off my video just for bandwidth issues, but I will still be here. So let me stop my video. And let's ask a really simple question to begin with. What are cell and gene therapies? I said that these are the medicines of the future. So let's define those a little bit more precisely. Cell therapies are exactly what the name implies. The cells are the medicine. We're using living cells to combat disease. Now, if you are undergoing a cell therapy and the cells that are being used to fight your illness come from your own body, it's what we call an autologous cellular therapy. By contrast, if the cells came from another person and are an off-the-shelf type of product where they've already been produced and QC'd and they're ready to go, it's what we call an allogeneic cell therapy. So these are two very important classes that basically all cellular therapies can kind of fall into. Now, there are a lot of different types of cells that are being used for therapy. I would say that this has been something that I've watched this bloom and blossom. But I would like to highlight one cell therapy in particular that you hear a lot about on the news. And if you go on the internet and you read stories, you hear a lot about what are called CAR T cellular therapies. And in CAR T cell therapies, what we do is we take T lymphocytes, which are a type of white blood cell, we isolate them from the blood, and we genetically modify them so that they're able to precisely target cancer cells. We put in a receptor called a CAR, or a chimeric antigen receptor, and that helps the T cells find cancer cells, recognize them, and kill them. Now, you can see below, I provided you with a list of some of the more recent approved cellular therapies out there, and you can see that so far the field has really been dominated by autologous cell therapies. The only problem with autologous is that sometimes it can take a little bit of time to produce the cells necessary. From the time that you're diagnosed and we collect blood samples to the time that we have quality controlled cells to put back into your body, sometimes that can be a lengthy process. And so there are a number of companies like Poseida, the Memorial Sloan Kettering Institute, and Allogene that are working on allogeneic cell therapies where you can go in and basically on demand, you can have a cell therapy. And we're very excited about these therapies because there are so many of them that are in clinical trials right now for a variety of different types of illnesses outside of cancers, including autoimmunity, multiple sclerosis, HIV, and things like this. So very exciting time in cellular therapies. Now, if we go to gene therapies, it's a little bit different. Gene therapies work a little differently because they're intended to create inherited genetic disorders. What we're in effect trying to do is enable genetic corrections where we go in and if an illness is based upon a dysfunctional gene, we simply replace it with a good copy of the gene and the disease is basically cured. Gene therapies really represent the closest that we can get to saying that we're going to cure a disease. We talk a lot about treatment and cures in the therapeutic industry, and this is really about the closest that we can get. Now, we can introduce those genes, which the genes are the medicines here. We can introduce those genes to a patient's cells through a variety of different vehicles. One of the most popular ways to do that now is through what are called adeno-associated viruses, or AAVs. And I've provided a list of some of the approved gene therapies that have already gone to market. And as you can see, there are things like spinal muscle atrophy that was a death sentence before, Duchenne's muscular dystrophy that I personally worked on cell therapies for. We now have gene therapies out there for that, hemophilias A and B. So a lot of hope is being brought to the world through these therapeutics. Now, if we take a step back and take a look at cell therapy workflows, there are obviously a lot of differences, but depending upon the different cell types that are being used, but they all share a few common features. Obviously, with a name like cell therapy, you're starting with cells. So you either need to have cells to start with, or you need to be able to isolate cells. Once you get those cells, you need to be able to grow them up to maximum numbers of healthy, high quality cells. And you need to be able to check those cells for their cellular identities as well as their physiological functions. You want to make sure that those cells are going to be capable of doing what you need them to do when they're administered to patients. And then the last thing is we need to be able, once we've produced those cells, we need to be able to freeze those cells down and store them until they're needed. As you can see, Thomas Scientific has been very hard at work in filling these gaps and really trying to bring value across the workflow. From cell lines that we have available to lymphocyte isolation kits that allow you to isolate cells. We also have supplies for growing up cells, including extracellular matrix proteins to coat your culture ware, microcarriers for suspension cultures, cell culture media, sera and serum alternatives, as well as recombinant growth factors and cytokines that we'll talk about in a little bit. We also have a number of tools that are available for the type of cellular analysis that is needed to really verify cellular function. And this includes antibodies and kits and sheath fluid to support flow cytometry. We also offer ELISA kits so that you can look at what the cells are secreting, the chemical signals that are given off. We have migration and chemotaxis kits. If your cells need to migrate to tumors, for example, we offer kits that can enable you to do that. Western blotting equipment, immunocytochemistry and immunohistochemistry supplies. And in some parts of the country, we can even fix you up with Leica microscopes. If these are of interest, reach out to your sales representative and you can learn more about them. Now, also when it comes to solutions for the freezing down and storage of cells, we offer everything from freezing media to keep your cells healthy during storage. We also have devices that are capable of freezing down your cells, including cool cells and controlled rate freezers. And of course, we have all of the consumable and laboratory supplies necessary to empower storage. Now, if we take a look at the gene therapy workflow, likewise, there are a lot of variations on this theme, but there are certain common things that we need to be able to do if we're going to make gene therapies. First, we need to be able to create plasmids that will help us to create viral particles carrying our genes of interest. We also are going to need things called helper plasmids that encode for things like the protein shells or capsids that surround viral particles and also enable viral replication. And we also need cells that we call packaging cells that can take all of these plasmids and basically combine them to create intact viral particles. Once we begin to create viral particles, we need to be able to efficiently and reliably collect and concentrate those particles, clean up any residual DNA from the host cells that might be floating around, and we need to be able to figure out the concentrations and the numbers of the viral particles that we produce. Now, here again, we at Thomas have worked very hard to fill these needs, and we've brought in vendors and vendor-specific products basically to support all of these different stages. Now, without going into all of the details, I will say that we have a huge variety of things to enable plasmid constructions. We have cells that are specifically bred to be able to robustly create viral particles in large numbers. We have efficient and optimal ways of getting plasmids into cells and also collecting and concentrating the viral particles when those begin to be produced. We're going to talk in just a moment about a couple of really exciting new products that we have for the cleanup of residual DNA. And then lastly, even though we're not going to talk about it in detail today, we have solutions for counting viral particles, including ELISA kits that allow us to count AAV2, 5, 6, 8, or 9 viral particles. We also have P24 ELISA kits to enable lentivirus to be counted. And we also have a device brought to us by one of our really great vendors, Corning, called the Video Drop. And this device allows us to actually see and to count lentiviral or adenoviral particles, not AAV though. Now, I'd like to switch gears for just a moment and talk about one of the very common problems that we encounter when we are developing cellular therapies. In the research and development stages, we are developing processes for expanding cells, maintaining cellular identities and physiologies. And if we're working on stem cells in particular, we need to be able to do directed differentiations or programming of cells. These all require recombinant growth factors that we can purchase from various vendors. And the processes are usually very, we test them, we retest them, we test them a third time and a fourth time, and we optimize them. And we're typically using research grade recombinant proteins since we're kind of messing around at this point, seeing what works. We're experimenting. It's what we do. We're scientists. However, when we begin to think about scaling up for clinical trials and we think about manufacturing, we need to think about not only GMP processes, which that stands for good manufacturing processes, but we also need to think about GMP grade reagents. Now, a lot of times in the past, that meant that we would have to switch from a research grade growth factor to a GMP grade. And the problem is if those aren't exactly alike, we may have to revalidate all of our processes again to make sure that the switch isn't going to impact our efficiency. Now, I can tell you that this can take a long time. This can take literally years to go back and figure out. So we want to be able to have solutions that overcome this problem so that we can seamlessly move from the research and development stages to the clinical stage. And we're very, very happy to partner with a company that I'll tell you a little bit more about in just a moment, where we're able to offer individual growth factors that are produced under either research use only, or RUO, or GMP auspices, so that they are basically, when you're ready to switch, you can literally seamlessly switch between a research grade protein and its equivalent GMP grade. Now, this company that we're partnering with is called Protein Tech. And this is literally right off of their website, one of their statements. And this explains a little bit more about what they're about with recombinant proteins. All of the recombinant proteins are produced and expressed in human cells. So they tend to be very, very high quality already. But the same expression systems, production processes, and operating procedures are in place for the research grade or the GMP grade recombinant proteins. The only difference is that the GMP grade are subjected to a little more documentation and a little bit more testing than the research grade are. Now, for many of the proteins in their portfolio, they already have RUO and GMP grades. If you need a particular RUO protein and it doesn't have a matching GMP grade, we also have the option of sitting down, you, me, and Protein Tech, and we can talk about actually producing a GMP grade and the tests that are going to be necessary so that we can give you a timeline on that. So what we're trying to do here is give you seamless transitioning between research and development and clinical studies. Now, this gives you an idea, and I won't go through all of these, but this is a list. This is another list of the research grade recombinant proteins, and then the GMP grade recombinant proteins. And like I said, anything that's on the RUO list, but not on the GMP list, we can actually work with Protein Tech and evaluate the possibility of making a GMP grade just for you. Now, I'd like to also take a look at a common problem that is encountered during AAV production, since we're also concerned with supporting gene therapies. Once we produce viral particles, one of the problems is that it's kind of a mess. We have viral particles floating around, but we also have a lot of DNA from the cells that we just used to produce those viral particles floating around. We want to be able to chew up those DNA fragments with specific nucleases. And the most commonly used nuclease for this is benzonase. Now, there are some problems with benzonase. At the optimal salt concentrations for benzonase activity, basically we have a lot of aggregation. The DNA and the viral particles clump together. If we add salt, they tend to deaggregate, and so we get more efficient degradation of that host cell DNA. Unfortunately, as we increase salt, we also see a decrease in the activity and the enzymatic ability of the benzonase nuclease. So we've partnered with a company called Arctic Zymes, where we have salt-active nucleases. And there are two different products here that I want to bring to your attention. The orange curve that you see on the right is for MSAN, and we also have SAN-HQ. Now, in the case of the MSAN, this is a nuclease that functions very, very well at higher salt concentrations. But really, the SAN-HQ actually becomes more active as the salt goes up. It's actually activated by the presence of salt. And so you can raise the salt, you can get rid of that aggregation, and your nuclease is going to function actually even better if you're using the SAN-HQ. And so ultimately, this gives you a little bit of a comparison. And you can see on the right how the MSAN and the SAN-HQ stack up relative to a benzonase or adenosine. So this represents a couple of the new products and kind of the new areas that we're moving into with cell and gene therapies. And like I said, we're trying to expand this portfolio to bring in the things that are going to help you to achieve success. I'd like to thank you for your time. Again, I'm Mike Moeller. I'm the Director of Business Development for Cell and Gene Therapies here at Thomas, and I'm happy to take your questions. Thank you.
Mike, I have a question. What expression systems do you use for making your recombinant proteins?
The recombinant proteins are all produced in what are called HEC293 cells, and those are human kidney cells. And the important thing about that is that when you produce recombinant proteins in human cells, they tend to work better on other human cell types. The modifications that they have when the recombinant proteins are being produced are more compatible. So in some cases, we actually see the recombinant proteins produced in human cells being a lot more potent.
Thank you.
Any other questions?
Thanks, Michael. Hey, I've worked with Michael, had him on a call with one of my gene and cell therapy accounts, and they're continuing to buy following that and continuing to follow up with Michael, asking him questions on their process. And so he's not really selling something, he's just showing them what the best practices, best processes are.
Mm-hmm.
And because of the knowledge that he's been able to provide them, it's actually given me some opportunity in some other accounts. So if you haven't, Michael's great to work with, and really has the respect out there of I know from my accounts, and especially those that are very technical, which this account is. They have a lot of good questions, and you can get very, very deep into it and lost. And Michael was awesome. So thank you, Michael. Appreciate it.
Thank you, Michael.
Sure.
I did see a question that popped up in the chat about the sources of the mesenchymal stem cells. And I'm really glad you asked that, because actually I am a mesenchymal stem cell guy. We actually have cells available from bone marrow as well as adipose tissue. And I'm actually working to bring in a vendor who's going to bring in placental mesenchymal stem cells. And we have media and growth factors that are capable of growing all of those. So I hope to be able to have a wide array of different mesenchymal stem cell types down the line. But right now it's bone marrow and adipose tissue derived primarily. I believe we also have umbilical cord derived, but I know that we're going to have placental and also some new umbilical derived lines coming down in the near future.
Excuse me, I just want to say we've just been notified by Zoom that there's some sort of glitch in the system, so we could be bounced out at any point.
Hi, Michael. John Montalvo here. Quick question.
Hi, John.
Could you maybe let us know some of the brands that we see in that workflow for the AAVs, just so that we have something to look for in the lab and know what's just most commonly being used, so we can speak to those products as well?
Yeah, definitely. I would say for plasmid prep, if you see a lot of Qiagen stuff sitting around, that's going to be a good sign. If you see Agilent stuff, actually a lot of our products for plasmids in particular come from Agilent. For the cell types, you may or may not see cells sitting around, but they should be using HEC-293 cells for that, and we actually have special cells for that that we have through Agilent. I would say that also, if you see bioreactor systems sitting around where they're rotating something slowly, ask them what they're making, because they could be making a viral prep. They could be making a yeast prep. It's always, I had somebody actually tell me recently they said, well, I went into a lab and I saw bioreactors and it had yellow stuff in it. What is it? And I said, well, I can't tell you what it is because, it could be a lot of things. So I think those are probably useful. I think the biggest way to identify people that are doing AAV is probably just to ask them if they're doing gene therapies.
I have a question.
Yes. Hello, Pat.
Hello, and Beth, you were absolutely correct. The second you said that we were going to get kicked off the call, I got kicked off the call.
Timing is
was able to get
everything.
back in. And I wish everybody else could hear this. As a sales rep, you're looking for people to talk to. What titles of people are you going to be searching for to hunt when you're out looking for these people? What is a title other than a PI?
Yeah.
What type of institutes would you look under? And I know it's to me, cell and gene therapy is very generic. Let's be a little more specific on,
sure.
Hey, this is going to be the so-and-so institute. Hey, this is going to be the so-and-so research institute. Can you give us a list of names to Google search? All right.
Definitely a lot of the cell therapies that are being developed now are for cancer. And so any place that is a cancer center or a cancer research center,
so
Fred
oncology.
Oncology. Fred Hutch up in Seattle, for example, Memorial Sloan Kettering, any place where, they're working on developing cancer therapeutics, ask them if they're into cell therapies. For gene therapies, I would say that it's, I would say gene therapies, they're probably going to say gene therapy in the name, or something that indicates that they are genetic oriented, or that they care about genes. Some of the titles of people that you're going to want to look for are process engineers. Process engineers are the guys who basically figure out, basically what you're figuring out the processes that are going to be used for production. And ultimately, this is where, your processes are going to have to go through research and development all the way up to GMP. You're also
what would their moniker be? Would it be, you know, like in chemical engineering, you have CHE? What would a moniker for a biochemical engineer be?
Well, you're not really so much a biochemical engineer. I mean, you know, again, the job titles are going to be like process engineer, process development. You're also going to have quality control people to some extent. Quality control, they don't always differentiate what they're quality controlling. But if it's a company that's known to be associated with cell and gene therapies, quality control people are good to talk to. Research scientists, senior scientists, these are the people who are actually doing the bench work. Unless you're me, I was a chief scientific officer that did all the bench work, or a good chunk of it myself. So, you know, when it came to the R&D stuff. So, you know, it's you know, I would say, you know, it's one of those things. If you have, I think probably, you know, your safest bet is if you have somebody that you suspect might be in cell and gene therapy. Chances are I know them or I've heard of them. Send me an email and ask me, and I can more than happy to take a look.
And I have a weird one for you. Would
Okay.
would veterinary hospitals be doing cell and gene therapy?
It's actually a really great question. Yes. The exciting thing is I actually, when I was at CRO, living in the CRO world, I actually did projects for veterinary stem cell therapies. And they were working specifically with osteoarthritis in dogs. And as somebody who owned dogs for decades, now I'm more of a cat guy, but you know, I owned dogs for a long time, and they were working on that. Also, horses, race horses. There's actually big business in cellular therapies. Gene therapies really haven't made it into the veterinary world. I would say that's more of a cellular therapy thing.
That's for our sales rep to go into College of Veterinary Medicines like I used to have to at Texas A&M. And I bet you nobody's talking about it, but somebody's doing it. So you just proved it.
Yeah, I've actually collaborated with a lot of you know, when I was in the biotech world as a chief scientific officer, I had numerous veterinary companies that approached me that wanted to partner with me to develop the same sort of platform that I developed for human cells. They wanted me to develop for racehorses. They wanted me to develop them for dogs, cats, you know, all sorts of things. And you know, it makes sense. I mean, mammals are really not that far apart physiologically and anatomically. We like to think we are, but not so much. More questions? All right. Well, guys, it's been a pleasure. Thank you so much for your kind attention, and I look forward to working in the field with you. And if you are a customer, I look forward to meeting you.
Yes, thank you very much, and apologies for having to jump off and jump back on. Hope you all have a great rest of your day, and this will be on demand within 24 hours, and I will put them back together. So if you have any other things, please feel free to reach out to anyone. There is a connect button up in the top left-hand side of the Thomas Center. So feel free to reach out if you had any additional questions.
Thank you, folks. Peace and blessings.
Thanks, Michael.
Thank you.